further. Not exactly sure, we're about 2 lectures short in this class, and I'm not really sure, but it's fine. I usually like to end up with some topics in biofilms and things like that, but I'll see how far we get today. Certainly we gotta get through the ARCHI, so that's my objective. So try not to linger too much in some of these. In the Tenuricides, kind of a phylum that perhaps doesn't spring to mind when we think about the bacteria, and we're still thinking about domain bacteria, is that is one of the groups, the Mycoplasma, and we have talked about these guys before. The smallest organisms, they are without a cell wall, and yet they're gram-positives. And this to some extent, at first, if we were traditional bacteriologists, we'd say, well, how can you do that? Gram stain is based on the nature of the cell wall. All of us here could explain that. I would think that'd be a reasonable outcome of our studies together this semester. And yet, it doesn't have that. It has this strange pleomorphic characteristic. And you could answer that by, maybe it's up here. So how do they do that? Why is it a gram-positive if there's no cell wall? And how unusual not to have a cell wall. Isn't that strange? How do they figure out that it's here and not someplace else? Why don't we just make it its own group? Oh, the Mycoplasma, let's just put those into their own little phylum. I mean, why not? There's already 39 of them. Let's make a whole bunch more. What are your thoughts? You can answer this. I'm not asking you something that would be like, oh, I don't know. I mean, you should be able to reflect on how we're doing systematics in modern microbiology and answer this question.
Is it a 16S?
Yeah, the 16S. Yeah, that's it. It was a short run for a short jump. That's kind of the thing we've really dominated our or started our attention on. And in that you should reflect, well, why the 16S? And we've talked about 4 main characteristics that make sense to us. So that's good. Well, as it turns out, as you might imagine, if you don't have a cell wall, bumping up to stuff in nature isn't a great idea for that particular group. And so we see many of them as parasites. Okay. And the other distinction we talked about, I think, really early, when we talked about what's big and what's small with the bacteria, we said, well, the Mycoplasma are the smallest ones, and here we are. And I always think of myco, that's kind of neat, fine. I think it stems off of some of the cellular characteristics, but the plasma is this kind of idea. These things are really don't have great shape. They're not rods, they're not spirals. And so we use the term, either here or one of my slides, it would be the term pleomorphic. I've used it before, but pleomorphic does mean that. Amorphous, or I think more amoeboid. Sometimes pleomorphic in bacteriology does mean that it will change shape. We've even said that you'll read in the Berkey's that E. coli is pleomorphic. But give me a break. Well, in the fact that when it's young, it's a young E. coli with many dreams for going out into the world and living its bacterial ways, it's a rod, and that's how we best think of E. coli. And yet, some of us from first-hand experience, once we let it grow for a while, it may become a cocobacillus, not oftentimes getting all the way to coccus, but really confuses the new student on looking at that, because your experience, you're still building experiments. Well, these guys don't have it. They are missing this cell wall. How unusual on that. So we look at this. This is even more shape than you're used to seeing with these things. Did I have a different picture? I did. I know on one of the early slides. Okay, that's it on. Yeah, this kind of thing. Okay, now these are a prepared sample. Here's a blood cell. It gives you some proportion. Wow, they are small. So that's a pretty good indicator of this pleomorphic state of these things. So small is free living. So we're still working on size, maybe. We look at the microscope. Oh, wow, those little bacteria, that's only 3 microns? Yeah. And these guys are about 1/10 of them. And you start saying, boy, they're almost the size of a virus. Now you're thinking. now you're on the right track, okay? And I think we've covered most of that, and I'll move on. Yes, the basic thing in the animals and plants. Mycoplasma diseases are better distinguished now, but for a long time they were hard to find. If you tried to look for them in culture, they're very small. Also, they didn't culture very well. Okay, these are fairly advanced colonies, and even those are eating into the agar. If you tried to filter them, they'd fall through the filters. So it was stuff. I mean, scientists got it, microbiologists got it, but it took a little bit more than the average. Oh, there's the rod, there's the coccus, kind of a thing. And also, wow, these things are small. All right. They're kind of talking about that. Yeah, they're related to cancer, related to other things. We're finding more and more as we study them, more things they're related to. But mycoplasmal diseases, I wouldn't say are rampant, but they are. There's a background level of them around. Anybody have any experience with yourself, family members, mycoplasma? Thought I might have stirred up some bad memories there or something. Okay, I think kind of cover, these are notes for you. I think we're covering most of the same thing. I'm going to skip spiroplasma. I think they're neat, but I love these kind of spirals. They're just the best shape ever for tunneling through your blood and your muscles, but we want to move on a little bit. So the actinobacteria, okay. I was surprised when I said, has anyone heard of the top 4, the most diverse groups? Several people said, well, yeah, I've heard of the actinobacteria. This is the first class that I've had that said that. Some people kind of heard of the proteobacteria, maybe the Firmicutes, but now the actinobacteria, so that's cool. That's really neat. So Corynebacterium, Arthrobacter, and Propionibacterium are the main ones. This is a fairly diverse family, so when we started out this whole thing, this whole discussion, several lectures ago, we had those big, whatever they were, trapezoids, the big colored branches of the tree of bacterial life. And we saw the actinobacteria, Proteobacteria, Firmicutes, and Bacteroidetes as the big 4, right? So to see that there's 30 families within the umbrella of this phylum does not really throw us too much, but it's like, well, that's quite a few families. Okay. Mostly commensals, variable for antibiotic production, and we'll see some of these, like the Propionibacterium, used in the dairy industry. So Corynebacterium, gram-positive aerobics, okay, these are the guys that are fairly common. Some are disease causers. Some are in the background of your flora. I'm not going to talk too much about Arthrobacteria, but here's Corynebacterium. They have this strange way of, there's something binary fission, and they kind of have this uneven fission plane going on with them. Okay, so all these, as you can see, well, these don't look the same, and this doesn't look like that. What's our term? Pleomorphic. Okay, it's kind of that big garbage bucket of terms we throw things in, like, well, it's not a spiral, it's not a vibrio, we'll call it pleomorphic. So they make them, you'll hear this described if you do a gram stain on these. I mean, gram positive, that's easy to see. But you'll hear them described as being club-shaped. And this is kind of, not like a club like on cards, it's like Fred Flintstone's club, It's kind of that kind of club kind of looking thing that they're talking about. And the other thing they'll talk about is their pattern, their arrangement. We'll say Staphylococcus is cluster-like, and Streptococcus is bead, beads in a row kind of a thing. And these ones, as I think I've told you guys in lab, those of you taking lab, that the clinical lab teachers will say, well, they stain in this kind of pattern of like oriental characters. Your eyes kind of search for a pattern, and I suppose if Oriental, maybe they're talking to you in some kind of bacterial way, I don't know. But they use that kind of idea of those characters as the pattern, so kind of like a pattern of no pattern is it, so, I don't know, is that really an Oriental pattern? I don't know that it is. I don't know why this isn't coming up. Okay, so, snapping division, you know, we don't talk about too much, like they will snap, you can't hear them, but you can watch them, they will kind of like start forming the wall and click apart. Acinetobacter will do that. They do that and they do some twitching motility, stuff we haven't talked about too much. There's so much more to go into. We'll talk about in a minute some other things. Erythrosma, the skin condition for Corynebacterium minuticinium is possible. So that one won't kill you, but it's irritating, like this button. And it will cause discoloration, some itching. The other one that we'll talk, I'll come back to Propionobacteria, it should be here. Yeah, the other one is diphtheria. Okay, Corynebacterium is known for that. Characteristic in this is this oral pharyngeal inflammation. So the diphtheria toxin not only causes bloating, but an inflammation of the neck. And there'll be a lot of gray mass in the oral, not cavity, the windpipe, okay? So that's a lot of dead cells along with mucus and so forth. So diphtheria can kill the cells and cause a fairly, well, it can be fatal. I mean, just not to mess with it. So sore throat, swelling. I mean, not every time you get a sore throat you have diphtheria. A few of you will ever, probably none of you will experience diphtheria, and I hope you never do. But I mean, if it was severe as with this child, you would know that you have something to look at, treatable, but nothing to fool around with. I think that's it for diphtheria. Yeah, the propionic bacteria I had listed here. In this group, we know that the propionics as being, one of the things we remember is that they help in the Swiss cheese. You know, you look at this, just like you look at the holes in bread now, you go like, oh, those holes in bread are from
Carbon dioxide?
Carbon dioxide generated by yeast. Yeah, so you go, oh, okay. I don't like always eating crackers. I'll put yeast in there. They'll put some CO2. Oh, I like fluffy things. Okay, so that's bread. When we look at Swiss cheese, it's got a characteristic taste. Some people love cheese. Some people hate cheese. I think if you're from Wisconsin, you have to like it, or they give you the gene. But to make you like it. But you go, oh, look at those nice holes in there. Okay, well, these guys are blowing open the cheese as it ferments with the holes. So in this group, we have that characteristic of Actinobacteria, which is kind of unusual when we look at where Actinobacteria is from. Another group worth talking about, probably talked about it a few times already, going back to the time of Cope, where we started all our lectures so long ago when the air was warm and the leaves were green. We had Mycobacterium. You said, well, does that relate to the study of fungus, mycology? We said, no, that's a good guess. It has to do with mycolic acid, and that, we said, well, you can learn the chemistry of that, but we have other things to do. Mycolic acid makes a real waxy cell wall. If you grow these things up, they really do. They look like little waxy bacteria. And we said, well, if you gram stain those, you're not going to get a reliable result. And if you go on into medicine, people will talk about, well, is it gram-positive, gram-negative, or acid-fast? Those are some of the big categories that you could dissect the Bergey's manual with. So we've talked about acid-fastness in lab. For those of you not in lab, basically you have to use a stain that you either steam in or have phenol in the stain. Or you have to, now we add detergent, we add a variation of Triton X that will permeabilize the cell wall enough to get a stain in there. You don't have to do that, as you know, with most of our gram-positives and negatives. Okay. I think this is it. People will talk about slow and fast growers. We will talk about, here's my colic acid. I know some of you are chemists and you love chemistry and you said, I wish we had more chemistry. And again, it's a balance act. But there is, if you're really interested in the stain that goes into really the fatty acid monomer, mycolic acid in there. Tuberculosis is one of the ones we talk about here. Mycobacterium tuberculosis. The cell wall is still being able to be penetrated. It does interact with antibiotics, so don't get mixed up that it's impermeable. I don't mean to give that kind of idea, but when we talk about tuberculosis, the thing about tuberculosis is, we see these background numbers. We go like, well, 26,000, 12,000 people die annually in this country. Worldwide, it's much higher. And we go, well, why don't we just give everybody antibiotics and make it go away? Well, for a while, and especially in our country, we're making good progress. I mean, tuberculosis, oftentimes we thought about as that disease of immigration in our country. I hate to be too general, but I think it's kind of fair to say that during the Industrial Revolution, when people were flocking to our country, living conditions weren't great, there wasn't central heating, people were packed together in miserable circumstances, not getting the best nutrition, and stuff like tuberculosis would take over. Kind of an echo of what was going on during the Dixonian England age that happened only a few decades earlier. So, that cough, that hacking cough, and hearing that people were dying from TB or consumption, was not unheard of. People didn't know what to call these things. People became slowly consumed, slowly started to die, slowly stopped to breathe. Although a lot of people could deal with it for a long time. You may or may not be having your lungs taken over quickly. Depends upon so many things, because we're dealing with biology. We're not dealing with like a chemical that would take over the body, a toxin. So, but TB is starting to show up more and more. Yeah, big numbers for other people. We're seeing it more and more, and this is that case of antibiotic resistance. Okay, it's a slow grower in general. We find mycobacteria like mycoplasma is related to other organisms. It can be missed. Different mycobacteria can be missed because of the degree of culturing. They don't culture necessarily fast, but you might want to use something like brain-heart infusion, which sounds kind of gross, but that's the media that they use for things that they call fastidious, kind of hard to grow things, and you give it just a whole bunch of protein and nutrients to make it grow. But then in our bodies, we take antibiotics, we might start to feel good, but antibiotics cost money, takes a lot of time, more money, let's face it, a lot of us don't want to take pills every day. Some of us are obsessed with taking pills, but some people don't do it. And then, of course, you're left with organisms that have seen a little bit of the antibiotic and not gone away, and those are the guys that hang out and become superbugs. A question I asked my nurses, or maybe I asked you, but no one bid at this, but how super can superbugs become? If we have antibiotics, we give them, and they say, well, they don't react to that antibiotic, we'll give them a different antibiotic. How super does it go? And where does it end? It's a question of evolution and adaptation that has no great answer, but has really scary consequences, right? It's like, well, if it's adaptation, unless you really get to some level of antibiotic, interaction, is there no end? Can you all just become so super? Well, the answer is, to some extent, you can only add antibiotics to such a level, and we only have so many different. That's not me, is it? You guys are hearing a bell? Okay. Okay, good, thanks. The, yeah, so, you know, we only have so many different kinds of bullets in the antibiotic gun against this war, and there's only so much we can put at so much level, because at some point, even antibiotics are toxic to the host or us. So it's a really interesting question with a lot of depth to it in that. TB keeps messing with us. So TB, you know, if you get a lung, a lung X-ray done, and you know you're not a smoker, you know, and you see something like that, you know, that's not a happy sight, possibly treatable. This is, not to make you feel really bad, I think this is from a deer. This isn't like some guy they just let. But basically, these nodules that you see is where the name comes from. These nodules, once they kind of calcify, they get hard, and thus the organ doesn't, as you might imagine, doesn't work very well. And they start to become tubercles, and thus tuberculosis. Yeah, I mean, other things can get it, you know, it's not just us, deer, raccoons. In this group, something that's really fun to talk about are the streptomycetes, and we'll do that next. That's one you're going to love, okay, I think. The actinomycetes, you know where we can go to find those? Right here. If any of you did any kind of gram stain or did something where you wiped your teeth, if we did that in this class, or you have time to do it, a lot of times you'll find a lot of strep, that's easy. Every once in a while you'll find these really thin little filaments, okay? And you go like, wow, it's almost like a fungal hyphae, it's so thin. That's usually these guys. They're really good at degrading a lot of different things, okay? So I don't think we're going to talk too much more about it, but that's kind of cool that they're showing up in our mouths. Nocardia is generally associated with sewage treatment, maybe a little bit higher in nutrients. Of the two species that now I'm thinking we'll never name for Central Park, both of those are Nocardia. We found probably about 50 new species, but it's dragged on now for about 8 years. I don't think we're going to get it done. But I'd like to. Did I tell you about the new species? Do you care? Do you want me to just move on? Anyone want to hear the story? Silly enough, Alex. Okay, really quickly. I had a sabbatical, 2 sabbaticals going on. I spent part of it looking at microbes. My question was, okay, only 1% of stuff can grow on plates, fair enough. Do we even know what that 1% is? You know, so we went in, and yeah, about 40% of the things we grow on plates, we can identify with GenBank. There's a whole bunch of stuff that we grow every day that we can't, that are like unknown species. You know, I thought this was clever on my part, maybe Kurt Suttle still thinks I'm an idiot, I'm sure it's true, but that was kind of a new finding. So we went in, and my student Mike Rossetto, tennis player here, actually, probably of some legend, but he went in and followed it up by doing sequencing, and to make a somewhat long story shorter, found 100 new species in Central Park, so we decided to take 2 of them and name them. And we got it down, you know, like I told you got to do the sequencing, you got to do the fatty acid, you got to do the hybridization. We did all that stuff, and it's hung up at the American Museum for different reasons. Anyway, we started thinking, well, what should we name this? You know, we didn't want to say, well, we don't want to call it Nocardia lemkii. That sounded a little bit presumptuous, although I think it would be cool. But so I decided that I'd name it after the microbiologist who started work at the museum. The museum's not known for microbiology, so Goldeneye, Professor Golden. And then the other one I was going to call Nocardia central ortis, which means Central Park, because I thought I could get in the New York Times and get famous. None of that's happened. Still here, famous in my own mind, right? But I thought it'd be cool for New York to have a bacteria named after him. You know, there's only a few states that have a state bacteria. You know, so thought that might be fun. Okay, back to task. So streptomyces is the one I really want you guys to know. These guys, for a long time, when we looked at them under the microscope, you would see them. You go back to the old textbooks, they classified these things as fungus. because they're very filamentous and they make a conidia, okay? Something we associate with aspergillus and penicillin, the molds, But now we know better. These streptomycetes are definitely in, this phylum, and they have a thing of their own. But something else, the conidia is another type of spore. Okay, so now, I don't know if we've done it yet, but we'll talk about myxospores, we've talked about endospores, and now we have these types of kind of conidial spores. So that whole thing is opening up. Oh, they're not just the same all the time? No, they're not. Okay, primarily in the soil. Have I covered the geosmins? I'm seeing blank faces. Let me go over it. So geosmins, you're experiencing these probably now. As you're walking to class this morning after a rain, we have kind of this whole new surge of streptomycetes in the soil mixing with these leaves, these little sheets of cellulose that are floating across the campus, brought to us by these deciduous trees. So the streptomycetes, as they degrade, will release, and I don't know if there's a function for this, but they release this smell that we associate with, okay, I'm not talking about old rotting leaves. Old rotting leaves smell like old socks, right? But that kind of smell we have, like that real earthy smell we might call it, or that fresh fall smell. You know what I'm saying? People who hunt, they use this smell to mask so other animals can't smell them. These are geosmins. Okay, that earth fresh scent. Okay, I don't know now if they manufacture, but that's what you're smelling is made by courtesy of streptomyces. Do you know the smell I'm talking about? That kind of earthy, autumn, nice rotting smell, rotting leaf smell. Anyone with me on that? Okay, good. Mike, what are you smelling? I don't know. What do you smell? Okay, but that's it. So, this is great dinner time conversation. Well, I'll tell you about those rotting leaves. People, my son and daughter are learning so much. I'm so glad that they have that crazy professor. So, streptomyces, yeah, they're very stringy-like. Like I said, think of them as kind of like mycelia, only in appearance, though. So, this is a neat thing. Now, one thing, and Dr. Veselnik, I don't know if some of you know him, but he was the clinical microbiologist here for years and years and years. Great man, still around doing stuff, but he decided to get off get out of the classroom and enjoy other aspects of his life. He said to me one thing that I've still contemplated on, and if any of you solve this, great. He says, why do, he's a mycologist, he says, why do fungus produce colors? They don't need it to reproduce. They're not drawing in insects or birds. Why is that? So we don't know. And sometimes colors are used to keep light away so you don't have mutation. We know that melanin and stuff helps with that. We don't know that. Do you know what happens?
What if it's like to help prevent them from being eaten by predators?
Maybe. For us it can work. We see something red, we go like, well, maybe I don't want to eat that. That could be. We don't know that. That's as good as an answer as anything. Okay, but so the idea of color amongst the fungus is up for debate. And maybe Eric will develop a thesis around that and become the next greatest mycologist dissertation or something. We don't know. Streptomyces produce a lot of different colors also. Again, don't know why. Just, it just seems to happen. But here's the big deal. 500 distinct antibiotics. You're like, well, why are we so worried about MRSA? If vancomycin doesn't work, we'll just find something else. It's not necessarily all that simple, but some produce more than one antibiotic because we are distilling something out of one plant or one bacterium, and we're applying it in an environment that may or may not have ever happened in nature. And that's fine. We're seeing something that works, and we're applying it. One must ask the question, as I've always asked. Well, you don't have to. One could ask the question. Ask what you want. Why did botulism produce toxins when it evolved so many years before? There's animals that even had that nerve synapse that botulism stops working. There's no nerves when Clostridium came around. You know, is it coincidence? You know, was there a purpose for a botulism toxin? I don't have the answer. In this case, we might think that, well, antibiotics, there might be a good adaptation for that, right? You got these guys growing in soil. What might antibiotics do to help streptomyces get a little bit of a gain on things? Produce antibiotics? Why would that help them? Or, no?
Producing antibiotics, you kill off other populations,
Yeah.
properties around you, you have more.
Right, it seems like that's a reasonable type of thing. I mean, just as we see some plants that'll produce a type of, black walnuts, okay, they produce a product that's high in humic acids and tannins, and eventually they'll just kill other things growing around their root base from grass. I was told I dropped. I think that the black ash, I call it the mahogany of North America, because its density and its beauty is like the woods we get from the rainforest. So I, of course, planted. I let the squirrels plant a whole bunch in my lawn, which my wife said, well, you gotta put them in rows. So I had to teach the squirrels how to put the nuts in the row. That took a while. But anyway, you know, my neighbors came and said, well, you reduced your property value by about 30% because black walnuts are going to kill your lawn, and people like to have green lawns. So we don't talk anymore. So I like black walnut. But that's the point. They do, you know, other plants will control their plants that way. So it's not a new strategy in nature. Streptomyces, you know, they're sitting out there with what? Bacillus. Lots of extracellular enzymes, a bunch of other stuff. It's like, well, if I can get you guys to move back a little bit, and some of you guys move back, I might be able to get some of these more dissolved nutrients for myself. It's a theory, and I think not that hard to make that connection. They kind of look like fungus. You know, I can see where guys are seeing them and saying, well, this kind of look like mold. You're going to have to get in there a little bit more and explore that. The top is supposed to have an antimicrobial. One of these is supposed to be, yeah, here. This one, it's harder in this light. Is pushing back on the growth of this one. So this is a streptomyces producing an antibiotic. Probably you can see it, I can see it better in here. You'll be able to see it better in your iPads and stuff, but that's the point. Yeah, here's these beautiful colors that show up. You can distinguish the different species both on their metabolites, but also on their conidia. You know, they're very varied. They're quite varied, is probably a better way to say that, looking at these. Who knows why? I mean, you know, why do we have some flowers that have umbels and other fruits that show up in racemes? You know, there's probably reasons for all of that, but I'm not exactly sure. But we have this kind of diversity, which under the microscope we can figure out fairly fast. Anyone do someone did streptomyces for their project, right? No, that's someone. Okay. So cool. But if you were to look at this, not very bacteria-like, you go like, well, you know, that's not like, you know, I'm expecting E. coli. No, yeah, you would now. You've taken this class. Well, I can tell you all about this. There they are. Really cool looking guys. And the chemistry, lots of antibiotic chemistry here, but 500 substances. The deal is, you know, people say, why can't we just find more streptomyces? Let's just go find them all over the place and see if they'll do stuff. And people are setting, well, actually that's kind of old news. First generation was setting up all these 96-well plates with different growth media and trying just to grow different species. Now you think, well, boy, this problem of only growing 1% is coming back to bite me, right? You know, if I'm more creative about how I grow things, maybe I can find the next new antibiotic. And then the next phase from that, then they got some success. They started looking at combinations, not combinations, trying to crossbreed these things, but combination of antibiotics that might work. And after that, it's a factorial design of complexity. You start adding more and more things that makes it really hard for the discovery. But believe me, people are definitely looking for this. You recognize names here that are probably very common to all of us. This is what we talked about. More, there's a nice one. See, this, what do you think this one is? We talked about that, the kind of feathery one, very common in soil. Okay, it's not a robin. Bacillus will do that. Bacillus, remember we talked about the molecular dialogue, the communication. They still set that up and it's kind of like frost. Bacillus. So this guy is actually knocking back bacillus, which is kind of cool. Okay, Bacteroidetes. Hard to say. Okay, before I gave my talk in Brazil, I actually downloaded the pronunciation and practice. I still messed it up. But Bacteroidetes is the way that it goes. Most people I know want to say, and I don't care how you say it in this case, but if you want to be cool, some people will say Bacteroidetes, okay, taking it more from the emphasis that you might use on a plant or a different type of Latin name. The museum people will say that. I'll say, no, it's Bacteroidetes, but who cares? Okay, let's talk about a few about these guys. I mean, did we really do the actinobacteria justice? No. Okay, let me just, before we start this, just in a little bit more in the actinobacteria. What we're finding now is a lot of diversity of the actinobacteria in soils. Okay, like the Bacteroidetes, we also find that the actinobacteria are located a lot in us and our intestines. I would say, if you had to line up the species diversity, your intestines microflora would not be all that different from soil, and probably even more similar to the types of things you might find in a composting heap. I'm not trying to say that for effect, but the types of things, the fermentations, the things that are going on are a little bit more like that. Now, of course, you're much more different. You have a lot of things going on, but still, if we have to think kind of environments outside of ourselves and similar diversities, that's what we'd see. Well, let's talk about at least the cytophaga and the flavobacteria. I don't know if you might get the stigmobacteria. But cytophaga, another one, people say cytophaga. I don't care about it. I looked that one up too because, I have nothing else to do. But I do enjoy trying to get stuff right. Where do we find this? Obligately anaerobic, okay? Many of them, numerically dominant bacteria in the human intestinal tract. Okay, probably, when I started teaching this, we didn't talk about this group at all. I don't know if it was even identified. I mean, it's probably a different name. But now we're seeing that more and more. So if you look at the human microflora, that comes up a lot. Some will produce sphingolipids. I don't know if we're going to talk about too much about that just because of time. Let's just see, sphingolipids. Let's go past that. So, cytophagales. Okay, long slender gram rods move by gliding. This is why I want to go back a little bit. Put on here many are obligate anaerobes, because the first one we start out with is cytophaga. It's not. So, but they're trying to be very general for the whole group. So put many here, and maybe I left it out. It could be my typo, actually. I'm not going to talk about sphingolipids now. You don't have a lot of time together. So, the cytophages. These guys are cool. You may have seen these. I don't know. I try to point them out. We didn't do a lot of exploration of nature, but we spread plated water. And you'll find these guys in water quite a bit. They move by gliding. Okay, remember gliding? And that's a different type of locomotion. Maybe look that over, especially if you're taking them to the comprehensive. They're not flagella. Gliding is kind of, putting out those protrusions and then, pulling forward on them and then pulling them up and having, almost like a little tank track kind of a thing, which is unusual because some gliding, like with the diatoms, they're not even sure how they even work yet. So, but bacterial clotting, we're starting to understand. Here's the deal I'd like you to remember, at least in general for the cytophages, is that they like to break down cellulose and chitin. This is why I was hoping, we had several groups breaking down cellulose. I was hoping we'd have little gold colonies. That's why I kept kind of pushing people towards, do a spread plate and put lens paper on it, because you often will see this time of year cytophages showing up in water. They get flushed in there, or they get a lot of organic, and they're breaking down cellulose, and it's pretty cool. So the most common natural polymer, the second most common natural polymer, okay? Beta glucose linkages, right? Beta-1,4, beta-1,4 with a little bit of nitrogen mixed in there. That's it. What a clever design. Okay, and these guys are like, yeah, pretty clever. I'm going to eat that. I think that's enough on that. Here's a picture of Cytophaga, probably the worst picture I've ever found on the internet. But anyway, I think we've been through most of this. Good way to think of these guys as they're really good at breaking down the natural polymers. You know, you go like, well, how does that help me? Well, when we think of Pseudomonas as being very diverse, good at breaking down anything from pollutants to other carbon, you know, the Pseudomonas are really the best at working on shorter chain aliphatics, straight chains. You know, they will tackle some simple aromatics. To break down cellulose, this long chain of beta-linked glucoses, you know, is harder. Usually, well, if we were in advanced micro, we talk about how you have some that come in and start popping off bonds at the end. Some will come in the middle. The pheneric fungus, for instance, and maybe you've seen this. You ever walk through the woods and you see a log that's rotting, and maybe you might see one of two things. You might see a log that's rotting and looks just like white paper. Like there's just this paper shell to it. Has anyone seen that kind of rotting? Maybe not. Well, if you see that, you have fungus and bacteria that have come in and they've eaten out all the lignin, all that epoxy-like glue, and they've left the cellulose. That's one group. Then, as you walk through life, you may notice a rotting wood, and it looks like a log that's rotting. You touch it, and it's crystalline, and it just all kind of falls apart. Okay, that's then maybe these guys, but also pheneric fungus that come in. They eat all the cellulose. They can't deal with the lignin, and that lignin, again, that epoxy glue between the vascular bundles is all there. You touch it, and it just falls down like a little crystal ice palace. You know, maybe I walk in the woods too much, but I mean, sometimes you see this kind of stuff, okay, in field ecology. Well, I think our group knows, you know, the difference in chitin and cellulose, but I put it in there. You know, its complexity lies in its minerality, you know, which gives it its structure, and its economy. I mean, we can break down glucose, we can make glucose in cells really well. It's a great idea, okay? But, you know, if we didn't have things that breaking down, as you can tell, year after year, if we didn't degrade these leaves, we would be walking through leaf tunnels, you know, to the class and stuff, so they definitely do their job. This is what you probably would see if you grew them on. This is what I'm most used to seeing. They'll grow, and then they glide. They'll start to spread on the petri dish. Not hard to grow, really. Definitely have an aerobic phase to it. That's why I appended that idea. Flavobacteria. Let's skip flavobacteria and sphingobacteria. I love talking about these guys. I'm just worried about our time. So, it's okay. It's okay. Flavobacteria, you'll see. You have them in you. You'll encounter those more if you study them. It won't be that hard to pick up. This is an interesting group. I guess they all are. I start everything that way. But let me tell you why these guys are interesting. Okay, so this group, the Chlamydia, the Planktomycetes and the Viromicrobia, something we found in the Amazon. It's around here. Crazy bacteria. We still don't know exactly what they do. Okay, I just published it. No one caught me on it, so I don't think anyone else knows what to do. That's what I wrote. So parasitic pore metallic capacities. Yeah, I mean in general that's okay. But they also are really good at breaking stuff down, but doing it in a simple manner. Chlamydia, STDs, right? I mean, gonorrhea, chlamydia, what else is in the top ones? I suppose to some extent HIV, but yeah, chlamydia is certainly up there and not going away. We have a background. I guess syphilis is always up there as one. Chlamydia, besides being an STD, it's a bothersome little parasite. It can also get to you by dry matter. Okay, sexual contact is a way of moving around and causing inflammation, burning, eventually dysfunctionality. But you can also have chlamydia showing up in other parts of your body. It can cause pneumonia. It can infect your eye. Okay, mucous membranes, open mucous membranes. You can get conjunctivitis from a trachoma. And then one type of avenue is psittacosis. Okay. It was showing up for a long time when people weren't aware of, people buy birds, right? Anybody have a bird? Well, some people buy birds, and they keep them in cages because they don't like them pooping on their furniture and stuff. So, and they might let them out and, teach them to talk and stuff like that. But anyway, when a bird poops, if they do have the chlamydial organism with it, sometimes what happens, okay, so if you clean it up, it's on your newspaper or whatever, not your hands, but it's on the newspaper, you throw it out, good. What was happening is birds were pooping, it's a high uric acid, it dries kind of fast, kind of like plaster, and so in cleaning it up, they're creating a dust, and not knowing it, they're cleaning it up, just, whistling a tune, and picking it up and infecting their lungs and getting sick. I mean, it can be very, very debilitating, okay, if left untreated. So that's in this group. Chlamydia. And of course, STDs, common, usually amongst, the sexually active people, usually the age group of what? I mean, people are sexually active before, but 18 to 25, you see the highest amount of this through experimentation and all that stuff that goes with that part of your life. Okay, this group I want to talk about more, okay? If you want to circle like Lemke likes this group, this is true. I have no doubt in it, because this one kind of, wake up now, stand up, turn around, get some oxygen in your lungs. We're going to talk about something that's kind of neat, okay? If you're kind of dozing, this is a good one to come back to earth with. Okay, some of these divide by budding, that's kind of cool. Some are stalker appendage. We've seen that before. Okay, this, look at this. And this is not, this is right. Extensive cell compartmentalization, including membrane enclosed nuclear structure. What? I thought you told me, Dr. Lemke, that prokaryotes don't really have a true nucleus. Okay, yeah, I did. This is like right on the edge. This is one of the groups where they're starting to say maybe there's some kind of pre-nucleus that came from these guys. Of course, there's the virus theory. There's other theories of how the nucleus established itself in the eukaryotic cell, thinking that eukaryotic cell came later. Okay, this one puts it out there, the Planktomycetes. Okay, there's some other cool things about these, and I want to tell you a few of those other things, and show you a few pictures. that membrane compartmental thing, that is pretty different. Okay, that really puts it right on the edge of, it's like when we look at the mix of bacteria. They have differentiation, they hunt other things, you know, that gets it right on the edge of, you know, why is it not eukaryotic?
Yeah, I was just gonna ask, how close is it related to eukaryotes?
Yeah, we're getting, you know, farther up the tree and more advanced things, yeah, but, closer, but it's still not. But, yeah. It still has the 16S, still has all that stuff. And, you know, and maybe, you know, when you're up here teaching people, it might be that we just started to reclassify stuff differently. You know, that's why I'm thinking the energy thermodynamic way of looking at things might be the way to think about it. I'm gonna write a paper on it, I'm sure it'll be ignored, but that's kind of, I'm thinking of the point. So we get weirdness with this. I've never seen these things growing. I've developed probes towards them because I was very interested in something that's coming up next, which I think is, here's this nucleoid thing. Isn't that interesting? Okay, it's only a single nuclear envelope. It's not double like your nuclei. But isn't that odd? Isn't that weird? So, I think it is. Jamata. Here's the deal, okay? This is another thing. In this group, over in either Austria or Germany, they started to find out, they're experimenting with, dealing with sewage is a big deal. It's $1 million industry, as you know, because everyone has to deal with it. Dealing with nutrient standards, getting rid of it, all that kind of stuff. It's not the most glamorous topic on the boardwalk, but it's certainly a necessary one. And until we had sewage treatment, you know, which showed up with the Romans or something, we were just dying right and left from disease. So it's worthwhile talking about. Now, these guys set up great big hemostat digesters, you know, flow-through digesters that were kind of a slower version of what's going on in sludge in a sewage treatment plant. And they started to find out that the nitrogen balance wasn't working out. Okay, you know, they knew about reducing nitrate to ammonia, you know, and they knew about denitrification, but, you know, a big part of the nutrient budget wasn't working out. And they started to find out that you had this process. I don't know if they first termed it, but this came out about 15 years ago. Maybe you're studying about this. Now when you study the nitrogen cycle, you should definitely talk about Animox because it's accounting for about 1/5 of the nitrogen shifting in the nitrogen cycle. Okay, if you don't talk about it, it just means that people don't get oxidative reductive conditions. Or they have other things to talk about. I'm not going to be too hard on people. Classes are complex. But here's the deal. Animox. It's kind of a pseudo-acronym. Anaerobic oxidation of ammonia. Okay, it's like, what? Usually anaerobic and oxidation don't fall into that you know, the same sentence, right? Oh, anaerobic, that's usually reducing conditions. Oxidation. So what they have, here's our ammonium. Okay, fair enough. Here's nitrite. Here you get then the oxidation of ammonia to nitrogen gas and the reduction of nitrate. This is your electron acceptor. No oxygen is in the formula. If we were to oxidize ammonia through regular you know, nitrification, we would have oxygen in the formula, right? So that's where it comes out. So it went under the radar. This is kind of hard to measure, although you did it with nitrate A and B in class. You can do it. I started showing students this. Oh, yeah, that's right. We can take something from microbiology and study stuff in nature. But 30 to 50% of this dinitrogen movement, you know, we're used to seeing this in denitrification. Okay, taking nitrate and reducing it to this, right? We'll go over those processes. Denitrification, nitrification, Animox. I would say I have a pretty good feeling for at least 3 of those ones for that. So these guys, Brocadia is the one we developed a primer for. One of the professor's sons came over. We did a science project. He probably went to state too. They started to find other ones. The Brocadia was the big one, which is in the Planktomyces. This is a big deal. This is like saying, well, you know, those archaea also do nutrient cycling. They're finding out that this kind of functional redundancy is important but not completely understood in nature. This is not even redundancy. This is a process that was going on under our noses that has just been brought out in the last decade or two. This is changing the way we think about nitrogen. And it came completely from the microbial stuff on working with anoxygenic oxidation is what's going on. Okay, this is just showing some more things. Here's a cartoon. Okay, this is the nitrogen cycle and where it fits in. I won't bust you on drawing this, but some of you would like to know, where would that go? Like nutrient cycles and that. Here's a cartoon. Here's that nucleoid we just saw a picture of. Okay, here's where this goes on. This is like another quasi-organelle that bacteria shouldn't have, and it's got the body of lysosome, whatever. So this is an anamoxysome going on in there. Okay, control the environment to allow this to go on. Isn't that interesting? Okay. I think so. Yeah, there it goes. Vermicrobia, just two things. One, they show up all over the place. They just weren't in the Amazon. You start looking at the river data, they're showing up. I think we even have them in us. Why not? What's interesting about these guys is here's kind of like pleomorphic to the largest degree. This kind of looks like those little, it's like a bacteria tardigrade looking thing. And I got these weird bumps and stuff on them. So that's about it. I would just say in your notes, the Vermicrobiota are under investigation, appear to be common, function still not clearly known, is what I would say with that. I think that's all I'm going to say about it. We got a few minutes. I think we can get to the end of the bacteria if you guys can hang tight for a little bit, because we're into the thermophiles. Okay. Now, thermophiles, we are talking at the base of the tree, right? We know the story, or at least the story we presented, so we're getting there. Here's Thermatoga. Okay, Carl Stetter was the guy they kept talking about. I didn't show you the part of that film, but Carl Stetter puts on this diving suit. It's really a diving suit. He jumps into a hot spring, and he's got this thermometer. It's like 150. I don't know how he didn't scald his stuff, but scald his body. But he's saying, yes, these bacteria do not eat pork. They eat sulfur and breathe no oxygen. He's a really cool character to listen. A real nice guy. But anyway, Thermatoga is the one we've talked about before. We've heard about before. Not at the record anymore. This used to be the record, but 90 degrees. The next time you see boiling water, take it off the stove, wait 10 minutes, and stick your finger in it. That's what these things are growing at. Okay, don't stick your finger in it, but you get the idea. Whoa, really hot places. They'll ferment chemoorganotropes. Share genes with the archaea. This is interesting for us. Some of you in the evolution question tackled that question. Okay. Related, don't worry about it. Since we're just kind of doing a tour, Thermatoga is fine for this group. Realizing other ones that have sulfur metabolism and live in hot places exist is the deal. Right? Not much to look at. Right? Okay. Aquaficiae. This is a cool group because we have Aquifex. Okay, and the Aquifex is the big one. This is mostly thermophilic, also growing in this group. This is a great one also at the base of the tree of life is Aquifex. Aquifex and Thermatoga are fine on that. Not much to look at. I want to talk to you about Deinococcus. Did we do that? Deinococcus and Thermosequana? We must have covered that because that's. Did we do that? You guys looking? Or just emailing people? Okay, you're smiling, I got it. Okay, well, I think we've covered it. This last slide, I won't read to you, but I wanted to tell, I find that people still mix up chemolithotrophy. So I went through and discussed it here. So read these slides. I won't read it to you, but if you have it, great. Good, move on. If it's kind of like, I'm not getting that chemolithotrophy. Lemke's talking about 5 different avenues of metabolism. It's not happening with me. And maybe at the last exam you saw that one diagram where I had that, oh, I went past that one. Okay, you know, it might come up, you know, so take a look at that. And then in the slide thing, hang on. Oh, there's your Deinococcus. I knew it was somewhere in my mind. This is how hot things are. We talked about that, but 121, the archaea, not the bacteria, still have the place for that. Here's Deinococcus and Thermus. Deinococcus is the one that can resist UV radiation. Thermus, or Thermus aquaticus, is the TAC DNA. We've talked about that. They're cool. I have Deinococcus if you want to grow it. I have this organism in culture. These are notable, but these are new additions. We're not going to talk about these in this class too much, so don't worry about these finally. The last thing I have in here, and it is time to go, so I'll let you read it, but I wrote down, we seem to be, as a class, some of you have it, some don't, I wanted to give you the notes I think are important for doing enrichment cultures, okay? And so you can read through this, and we can talk about it next time if you want, but I still feel that people aren't getting the idea of enrichment. The way that Bajornik and Winogradski really started thinking about it, and then so many others after that. Enrichment culture opens up the world of the unknown to you. You can grasp it by enriching and thinking about the environment these things grow in. It's your spacesuit into this world. So thinking about it is worthwhile, and I'll give you some examples. Okay.